Welcome back to the Business of Biotech. I'm Matt Pillar, and today we're talking about the intersection of biotherapeutics and medical devices with Daniel O'Connor, President, CEO, and Director at OncoSec Medical. Now, Dan brings an interesting combination of experiences to his role at OncoSec. He's a veteran of the United States Marine Corps, where he was among the first U.S. Marines deployed to Saudi Arabia for Operation Desert Shield before attaining the rank of captain. Dan, before I go on, thank you for your service.
Matt, thank you so much. You get more than you give, and being able to serve, in the Marines, I got so much more back than what I gave. So it was really, an awesome opportunity and a privilege, actually.
I'm sure it was. And I have to tell you, before I share with our audience a little bit more about you, I was not to date you, although I already have by mentioning that deployment. I was a sophomore in high school when that was occurring. And I have to tell you, it was one of the, it was definitely the first vivid, the first vivid experience for me of our participation in a global conflict. And I can remember, watching the news footage of the tracer fire and just all the activity that just seemed to happen right now overnight. And, it's, like I said, it left an impression for sure about the sacrifice that guys like you made back then. And we really appreciate it. Yeah,
you bet. And again, I've gotten so much more of the service of the Marine Corps than I ever gave to the U.S. Marines. I gave it everything I could have. But it's a great life lesson that you get more than you give. When you give, you get in return, and kind of the more you give, the more you get. Really is kind of the philosophy there. But I had those. I think that was the first time wars were actually televised, if you will, and the Patriot missile, going off, and those protective missiles that were going against the the bombs that were being launched against Israel and elsewhere. I think it was really dramatic and kind of brought it into everybody's front and center. And I agree with you, very memorable time.
Sure was, yeah. So moving on from that, you earned your Penn State Dickinson law degree. This was post service, I'm assuming.
Yes.
Yep. And then Dan served as a prosecutor for a while before jumping into the life sciences. He served as counsel for the CRO Inventive, SVP and general counsel at ImClone, which is an Eli Lilly company, and VP and general counsel for the med device company Bracco Diagnostics. So a lot of legal experience there before jumping into immunotherapy leadership as CEO and director at Vaxxus. In 2017. Dan took the helm at OncoSec, which isn't just developing immunotherapeutics to treat cancer. It's developing a proprietary delivery system for those therapies, and that's one of the things we want to talk about today. In the meantime, Dan also serves as vice chair and vice chair at Bio New Jersey, correct?
Yeah, the board of trustees. Actually, we just did an election, and I stepped down as my role of vice chair. I'm still on the board of trustees, but.
Okay.
Kind of handed the baton to another great person to run that, to be the vice chair. And then we have a new chairman as well. So it's excellent.
Excellent. Yeah. Very important organization, very active on the scene. So thanks for your service there as well. You were also an appointee to the governor's New Jersey Biotechnology Task Force not too long ago.
Yeah. That was the prior governor in New Jersey set up a task force for biotech, and asked me if I wanted to participate. And again, I was so pleased to be able to participate because it was a great experience. And it just kind of highlights the role that New Jersey plays in drug development. And it's a really profound role. Some folks call New Jersey the medicine chest of the world. And I think that's in a lot of ways very accurate, because many of the drugs that get approved every year originated or have some type of footprint in Jersey. Mm-hmm. So it's good. It's a great place to. I've been extremely fortunate to have been able to make a career here, developing drugs. So it's again, you get more than you give. You work hard to try to develop drugs for people that need them and need better solutions, especially patients that have cancer. It's been extremely rewarding. And thank you for going through that. I appreciate it. I actually felt a little tired when I listened to it all. But that's good.
No. I mean, you've been at OncoSec's since 2017, so going on 4 years. You've still got plenty of work to do. Don't get tired yet.
Yes, more tread on the tire.
So I want to talk about your professional background a little bit. I mean, it traverses med device, biopharma, obviously law, and the military for that matter. That combination seems like a killer combination to step into a leadership role with a company like OncoSec's. So, I mean, tell me. I know it's a big, broad, open-ended question, but give me your thoughts on that. What aspects of that professional background have best prepared you for the role that you serve in now?
Yeah, so I think it's just tying back to the Marines. The Marines teach you leadership, and the leadership is leadership by example always. And I was really fortunate to be able to make it through a program called Officers Candidate School and be being able to be an officer in the Marine Corps, and then you get the privilege of leading Marines. And the Marines motto and the Marines, kind of leadership training is, in the bedrock of that is everyone else first. The people that you're leading come first. You come last. When there's a tough job to do or something people don't want to do, then you are the first one to go. And that's leadership by example. But it's, the servant leader, if you will. I think that's the concept. But it's really putting the Marines that are working for you up first and certainly above you. And then never not being willing to do the dirty jobs that need to be done. I think that was, has always, been something I've worked to do. And, in whether I had, one person working for me or, 100 people working for me. It's, always trying to be that servant leader and make sure people understand that you care about them, that, their career is important to you, not just what they're doing for the company and adding value to the company, but in fact, what their career development is, their family, pretty much everything. And, that I think that leadership is, for lack of a better word to describe a program, if you will, is the best one. And because, the ultimate goal is when you're a leader is, having people, follow you and do that willingly in a way that they're happy to do it, kind of willingness to follow. And, again, when you're taking care of people and showing them that it's them before you, or if there's a problem, the finger gets pointed to me, right? The accountability part of it, it's not a blame, shifting blame. There's always accountability, like, hey, you didn't do that right. Let's fix it. Let's do it right the next time. But the ultimate responsibility and problems when things go wrong are at me. Again, the Marine Corps accountability, piece. But I think that was, kind of the biggest. The other, so again, kind of making sure that the people that you're working with and that are, working for you understand that they come first before your needs. And the other part is making sure everybody understands the mission, right? One of the things you're taught in the Marines is what's called commander's intent, but which is the idea that everybody in the organization knows what the objective is, from the person who's at the lowest level to the person that's the top of the hierarchy. And that person at the top has to make sure they're effectively communicating that throughout the organization. And, there's a lot of reasons for that, but the one I like is when you're in the workplace, people are making decisions every day. And, if they understand the ultimate objective, it's definitely going to help them make the right decision, because you're not always going to be there to help people make decisions. And you want to foster that good decision making. When they understand the bigger objective, then they can make better decisions. And I think it also has everybody feel like they're inclusive or included in the organization. They're not just kind of a cog in the wheel, but in the machine, but they understand the bigger picture, and they understand it from the leadership. So I think, that helps, with morale and with kind of, again, people feeling that they're part of something bigger than their role. But it also, fosters the opportunity for people to make smart decisions when they have to make a decision, if they know where, we're ultimately driving for.
Yep.
So anyway, that's, again, a Marine Corps thing. You're always teaching people what the mission is, what the objective is. And, that's the person who's, the private at the fire team level, all the way up to the top.
Yeah. So I want to ask you this, we could talk about leadership and that discipline for a long time, and I do want to be conscious of our time and transition to OncoSec. But I have to ask you this: so you're not the first lawyer turned biotech CEO that I've interviewed. It's fairly common, and I can absolutely see where that law background really plays nicely into this business, because there's so much, legal consideration, patent law, so on and so forth, that I'm sure you're very well versed in. But your undergrad is in, I think, English or literature. You served in the military. You attained your law degree, and you've worked in biotech for quite some time. But has the lack, obviously you're diverse and very well read, very well studied. But has the lack of the lack of scientific background, I guess, or by education or training anyway, ever felt like a handicap?
No, great question, and thanks for asking it. Actually, I wanted to be a doctor in high school, and I didn't think I was smart enough to do it, and so I didn't. I focused on an area that I kind of excelled in, which was words and reading and writing.
Yeah.
But I wanted to be, I knew that was something I was gravitating towards. And I get, everybody is wired a certain way. And the one thing I'm a big proponent of is understanding how you're wired early in your career, right? You can take a personality test at the end of your career and find out you went through all these different stages in your career development, but you ended up to be the place that you were wired to be in the first place. So sometimes knowing that out front is not a bad idea. But I don't read. I don't read for pleasure. The reading I do and the teaching I've done, since getting into drug development, has been to learn the science. And when I started at the CRO, which is what's called a contract or a clinical research organization, actually I was very fortunate to be a founding employee of a company called PharmaNet, which then became Inventive Health, as you mentioned up front, and then is today Syneos Health, which is one of the largest CROs in the world. And it's a very, very substantial organization. It had its genealogy in this company called PharmaNet, which was a startup CRO. And CROs actually it's kind of an outsourcing model for clinical research. And clinical research is really, at its essence, running a clinical study in a doctor's office or in a hospital. And so pharma companies used to run those internally, and some still do to some level. But the model that actually was developed by the gentleman who founded PharmaNet, he also founded a company called Covance, which exists today. It was called Bestlar Associates a long time ago. Yeah. In this concept of outsourcing clinical research. And I was a lawyer, transitioned from prosecutor to PharmaNet, and I was very lucky to be able to wear many hats, not just a legal hat, but I got in charge of facilities. I then moved into identifying the financial progress on our projects. But in all of that, I had the opportunity to study the science of each of the projects we were working on. And, had a lot of gracious people around me that would take the time to explain to me what the drug was and how it was thought to work. And, in that, I got exposed to a tremendous amount of technology. And again, had the ability, when I read the contract, to go through the contract, I would read the proposal, and the proposal would outline the scientific premise of the drug, the product candidate being evaluated, and I would learn that way, and I could, again, would ask questions. So that was always kind of a grounding learning that I started from 20 plus years ago. And over those years, I was really fortunate to be able to migrate towards oncology, and in particular, immunology. And at ImClone, we developed a drug called Erbitux, and several other of the drugs that were designed in ImClone, they're monoclonal antibodies, eventually got drug approval by Eli Lilly when they acquired ImClone. And at ImClone, the antibodies that we were working on and our scientists were designing had an immunological aspect to them. They were basically blocking monoclonal antibodies that inhibited a part of a cell's growth. In the case of Erbitux, it's the epidermal growth factor receptor, which is a receptor on the surface of a tumor that when stimulated or when activated will cause tumor expansion and growth and vascularization of the tumor. So that antibody, had an immunological aspect to it into a certain way. And there were immunologists at ImClone that, started teaching me, and I asked them to, start to teach me. And I got their textbook from school, and I started to study it. And that, from my point of view, I was yes, had a legal background. I was fortunate to expand that to a lawyer business partner role. Really kind of gravitated towards, but spent the time learning the science. And now I could probably really teach the first 2 chapters of a immunology book. Beyond that, I pretty much it gets over my head.
Sure.
But that grounding is critical for a CEO in this business, because a lot of times you see doctors, right? And then you thought, which is an appropriate thought, which is, if you're a lawyer, you're not a science guy. And it's true, the formal science training is definitely not there. But I think in this business, if you don't have some type of hunger for the science and natural, wanting to learn it, it's definitely, you may not be as successful as you hope to be.
Sure. Yeah.
Because the other part is you get pitched ideas a lot, right? And our scientists are constantly thinking of things. And, ultimately the decision to move forward with something is going to be, it may rest with you and your board of directors. So if you're not able to kind of connect the dots in the science, it gets hard to make those decisions.
Yep.
At least having a grounding is helpful. So anyway, hopefully that answered your question. I mean, I eventually got to the point where at Evaxis, I was an inventor on some of the patents there. The idea of using a neoepitope as a target for a cancer immunotherapy was something I envisioned, through a dialogue with our then CSO. And, and that was, again, kind of one advantage you have as the layperson is you can kind of after a while start to piece it all together and marry up the business opportunity with the scientific premise.
Sure.
In that case, it was, one of the problems with cancer immunotherapies is they're promiscuous. That when you get a cancer drug, is it designed for you, the person? No, it's designed for the masses, and it's studied as such. But a better approach would be to develop a cancer drug that actually is designed for you. Mm-hmm. Cancer is a disease of mutations. Mutations are specific to individuals. We now, through the work that's been done to identify the human genome, we're able to relatively quickly, interrogate a tumor and find out what those mutations are. And having that foundation and understanding that an important business / commercial need / patient need is to identify a cancer drug that's not built for one size fits all, but instead targeted to the individual, is a very important thing.
Right.
How many times does a cancer patient hear when they're told how, the first question is a drug going to work when they're being told they're going to get a treatment? And then they get percentages. Right? Well, 20% of the patients, 30% of the patients, right? I mean, how much better would it be to say, look, this drug has been designed for you. And, it's highly likely to work because it's targeting your mutation. I guess that's the ultimate goal. But kind of, again, having that business background and then learning the science to a certain extent is kind of helpful to bring those things together.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech.
All right, so let's shift into how that's playing out at OncoSec. And I wasn't sure when I was putting some thought into this conversation. I wasn't sure whether I wanted to start with the therapy or the delivery mechanism, or if that conversation should happen in tandem to sort of describe the relationship between the therapy and the delivery mechanism. So I'm going to leave that up to you, Dan. You tell me. I want you to give us an education on what OncoSec's doing. Yeah. Does it make best sense to talk about those things in tandem or separately?
Yeah, it's a great question. And believe it or not, when we talk about technology, I kind of do that by the person I'm speaking with sometimes. But I actually want to pause and thank you for doing so much homework, Matt, about the company and about me. I really appreciate that. It's impressive. And, I definitely appreciate that because you're really hitting on, when we talk about our technology, we're teaching it to people.
Yeah.
And you obviously want to teach it in the best way possible so that they can understand it and appreciate it. And then again, manipulate that information for themselves. It is a drug device combination, and the drug itself is a biologic, and it's an immunotherapy, which is why under our name, you can see me the sign behind me, or actually not this one, but our logo, we put the words immunology. That's using the body's own immune system to attack a cancer. In that field of immunology, the field is always looking at things to communicate with the immune system in some fashion. And, and we use all sorts of tools to do that, because, the premise is if a patient's got cancer, you need to eradicate the cancer. Well, how do we do that today? The mainstays are chemicals, chemotherapy, radiation, radiotherapy. And now immunotherapy is egressing in, and targeted therapies, which was the clones, Erbitux and other drugs that are like that. So all those therapies, especially chemoradiation, come at a pretty high cost to the patient. And we all know what those are. We know people have endured or had to endure chemotherapy. They're great drugs. They save lives, but they have an impact. And the concept for cancer immunotherapy is to avoid those consequences and use the body's natural immune response to eradicate the cancer. That's the underlying premise. The challenge is talking to the immune system, because when a patient has cancer, the immune system gets basically tamped down or shut off. And so you're trying to reactivate that immune response. And there's lots of different tools to do that or interact with that immune response. Bacterias, we already used with Advaxis, it was Listeria monocytogenes. Why do you use a bacteria? The immune system recognizes it, eats it, gobbles it up, and you can communicate with that connection. Adenoviruses, there's drugs that are adenoviruses, and they're same concept. They will interact with the immune response because your body recognizes a virus. Monoclonal antibodies, they're manipulated to mimic the shape of a protein and inhibit some aspect of the immune response. So there's the communication link. In our case, we use energy. And energy is, what I really like about that is it's, when you're thinking, again, the big premise is to get away from the side effects of chemoradiation, not to supplant them with new side effects related to immunotherapy, right? So, it is moving we want to move the ball further for the patient, right? And doing that and really kind of shifting from one type of side effects to another different type of side effects that still have pretty significant consequences. It's moving the ball, but it's actually not getting to the goal, which is to get away from those, the things that are the cytotoxic side effects of, drugs like chemo and radiation that are killing all the cells, and really try to figure out a way to not supplant them with immunotherapy side effects, but really communicate with that immune response in a way that's going to avoid those side effects. And I think that's one of the things that is kind of it relates to your question, which is our device part of our drug combination, because that uses energy to communicate with the immune response. And in essence, what we do is we take a strand of DNA, which is called a plasmid. A lot of people know about immunology now because it's hugely important with COVID-19, right? If you took a poll before COVID came into being, you'd probably find maybe 1 out of 10 people knew what the word immunology was, or kind of had a sense of it. Today, people are pretty well educated on this topic, right? Well, people now understand DNA. Moderna's drug is RNA, right? It's an RNA approach. Well, DNA and RNA are, related. The DNA is basically blueprints that tells the cell to do something. And in all those other systems I described, in many of them, they use this plasmid approach because you can code CODE onto the plasmid things that you want to embed into cancer cells and get this cancer cells to make using those blueprints, right? So we make that communication and that process of getting the DNA into a cancer cell using energy. Energy is effervescent, right? It really has no physical form. It dissipates in a second, but it achieves the goal of causing a cancer cell's membrane to become porous, or a tiny micropores form for a moment. And we've previously injected into the tumor billions of copies of the DNA. So billions of DNA plasmids coded for something known as IL-12. And so the combination of the drug and the device is really important from our standpoint, because efficacy is the single most important thing people focus on, and rightly so. Is this drug going to work for me? The next question is, what's it going to do to me, right? What are the side effects? Mm-hmm. And, when you can have that risk benefit, the benefit of the drug being high and the risk of consequences, side effects being low, you really start to, generate an exciting thing. And in our case, one thing that gets overlooked, I mean, you identified it right away, which is the device component to us. People that kind of misunderstand that part of us a bit. But it's really not driving the efficacy. The DNA, the biologic part is really what's causing the tumor to shrink. But getting that DNA into the cell in a way that actually doesn't cause the patient experience to be more harsh, again, supplanting immunotherapy side effects, from chemotherapy side effects, is this system from our standpoint, which is we feel is underappreciated. And it also sounds a little like old school. Like you're sometimes people say, well, you're zapping tumors. It's actually not really true. What we're doing is we're providing a small amount of electrical energy in cancer cells and millions of cancer cells located in a tumor to keep them alive. We don't want to kill those cells because we're going to use those cells as factories to make our immunotherapy. Mm-hmm. And in our case, immunotherapy is causing the immune system to kind of reignite, if you will. It signals inflammation. Yeah. And it ends up dovetailing very nice with a class of drugs called checkpoint inhibitors, which are very important today. So anyway, hopefully I answered your question, Matt. It was probably a little bit deeper than you were hoping for. Hopefully I didn't chew up too much time on that one.
No, that's good. We definitely wanted a thorough understanding of that, and it leads to probably a host of questions around manufacturing and regulatory considerations that OncoSec has had to tackle. Because it occurs to me, and again, I'm not a scientist, but it occurs to me at a very simple level that this approach, in effect, doubles your manufacturing considerations. It doubles your regulatory considerations. Yes. It's not a straightforward like, hey, here's our BLA. We're gonna, we've got a biologic therapy. You're tackling biologics and med device in tandem. So, yeah. I guess I'd start with the company's approach to manufacturing. Lay that out for us. Is there a sort of a separate like device manufacturing effort happening, separate to the immunotherapy manufacturing approach? Do they happen somehow in tandem? I don't.
It's another great. Let's do again. Thanks for studying the company. Question. There is an additional layer of complexity, right? Mm-hmm. But on the other hand, the trade-off of not using a bacteria, a virus, some other physical things, definitely worth it, right? Because we deliver a DNA plasmid. That's all we're giving, which in and of itself is inert. We're not wrapping that in a lipoprotein. We're not wrapping that in some kind of thing that gets the immune system to tackle it. And that creates that communication link that we're looking for using that virus, because the immune system recognizes the text virus, nanoparticle, et cetera. All of those are other added elements that have to be brought into the picture of the drug to make the drug talk to the immune response. We use a different tool, energy, and I like it because again, when you look at the risk benefit, the benefit is looking good. The risk is, we have a very, very what's called clean safety profile. It's very few treatment-related adverse events related to the technology, and that's because we deliver it to the tumor, but we also aren't using some other physical thing that the body has to do something with, like a bacteria, which may cause your blood pressure to drop when it goes into your bloodstream. It's the trade-off. So in my mind, adding that layer of complexity for the device is absolutely worth the trade-off, because you're getting that, that ultimate goal of not supplanting one form of side effect with another. In our case, actually, is end up being a reality, and it's all attributed to this device part of us, which in and of itself has no efficacy. It's not causing the patient to get better. Right. Yep. It's like a syringe in a way. It's like a tool that's delivering this energy. And what's also nice about it, and what I like about it, is the plasmid side of the manufacturing house is very simple, right? Again, not using some other kind of thing, nanoparticle, virus, bacteria, antibody, that has a very relatively complicated manufacturing process. Plasmids can be made in bulk and they're made quickly and they're stable. So that part of our manufacturing is very simple, straightforward, and low cost. The device itself is a fixed cost, right? You make that device, it's made, it lasts for years. The thing that needs to be changed are the applicators, and those are disposable. So, so that is I think on balance, when you aggregate the two, our manufacturing process, although kind of at the surface would say you got two things you got to go through here and two processes that have to occur. I think when you weigh that against what you're achieving with the device versus what you have to introduce on the drug side to get to the same result, it's absolutely worth it. And a lot of it also is, the engineering. Once it's done, right? Once it's reviewed, right? You can make improvements to it, and that process needs to get reviewed. But it's, once it occurs, it's done.
Sure.
Anyway, sorry, hopefully that answered your question.
It does, yeah. Now, when you faced up with the regulatory agencies, were you approaching the FDA in tandem, or like with the immunotherapy and the device, or did these efforts happen separately?
Yeah, it's reviewed as a combination product, but you have to review both parts of that puzzle as well as other parts. But just the biologic side of it and the device side of it both get reviewed and have to be cleared. And in order to conduct what's known as an investigative new drug, or an IND. And that process to get the IND, you provide all the information with respect to both pieces, the device part and the drug part, and those both have to be reviewed and cleared to be able to move forward into human clinical trials.
Yeah, given that the device aspect is not a traditional, sort of like a traditional syringe, for instance. Was that considered uncharted territory by the regulatory agency, or was there some precedent for that?
No, they have a whole group that works on devices and with devices. A lot of those devices have a therapeutic effect themselves. They do something therapeutically. Right? They're delivering a benefit to the patient. They're doing something. In our case, it's not. It's delivering energy. You could do that in a tumor, and you're not going to get any kind of anti-tumor activity. You're just going to see dispersal of energy into a setting. So, there's a whole device component to FDA's, the reviewers, and, they look at all devices, but there's also devices that have, a therapeutic effect. And that's like judging the biologic part of it within the device. Is it doing what it's designed to do? Is it having the effect it was designed to have? In our case, the device doesn't have a therapeutic effect. So it's really just looking at the safety of the device, the engineering of the device, making sure that it operates the way it's designed to operate, and it doesn't expose the patients to risk.
Yeah. Okay. Were there any particular, as you described, the therapy itself is relatively straightforward. Were there any particular challenges that you had to lead the company through in terms of manufacturing? And if so, how did you go about that?
I would say not so much manufacturing. I mean, it's the device is being evaluated. We have one in clinic. We've got one that we're developing for commercial. So that's being evaluated right now. We worked with our COVID-19 vaccine with one of the very few companies in the United States that has a product that's being evaluated in a clinical trial for COVID-19. And that we in-licensed a device from an Italian manufacturer. With this device is actually their device, and now ours, exclusively in-licensed in the United States, actually worldwide, is used worldwide. So it's been approved in Europe. It was not used in the United States, but when we moved forward with an IND for a COVID vaccine, called this Corvax 12, because we use IL-12 as our biologic, we had to get that device cleared, and it took months, but it happened, which is great, and it's now being used. I think that the bigger, kind of obstacle, hurdle, if you will, or challenge is, people that haven't used it before, right? So you have to train people on how to use this. Now, the training is not that hard. It's, there's a slide in our slide deck which shows you put a syringe with the DNA into the tumor, and then you push the applicator into the tumor. You depress a foot pedal, and it delivers the energy, and you remove the applicator. It takes about 3 seconds to do, and the entire patient time is about 10 minutes. So it's a very quick, easy approach. But people that, it's when people have never used something like that, most cancer doctors write a prescription and send a patient to an infusion clinic, right? That's a different, workflow for them.
Yeah.
And, and so there's a bit of getting, investigators on our clinical studies, interested in using the technology, and then using the technology. And I think what we've seen is, some of the, obstacles coming into it, or maybe biases that people have vis-a-vis the technology, erode, once they use it. And then when they see the results, then they become, strong proponents of it, which I think has continuing to happen for us.
Yeah.
Which is great. I mean, it's a process basically, and it is a challenge, getting a study up and running and opening sites and training those sites and ensuring that they're comfortable using the technology. There's not a lot of art to it. It's really what I just described. But going through that process is not something that's typical. And whenever you're bringing the non-typical in, it's you've got things you have to work through and deal with.
Yeah, requires a little more field work than simply shipping vials. Yes,
absolutely right. And it's way different than putting a vial in and hanging a bag and putting a line into a port. It's a process. But what we do with that is we have training videos. Pre-COVID, we would have people, we call boots on the ground, but people in the doctor's offices, interacting with the site staff and teaching and learning in a way too, from their experience and techniques and things like that. But with COVID, it became a little bit more challenging. Everybody's doing this. So, but I think the good is that, all the training we do, yields the result we want, which is people properly using the technology and, hopefully getting results for their patients.
Yeah. So let's talk about that a little bit. People properly using the technology, and there's a lot of that going on because you guys have a lot of clinical activity happening. I mean, you look at your pipeline. There's a lot happening. So we obviously don't have time to drill down to every single one of those programs, but I want to give you an opportunity, Dan, to share with us the key programs that you're working on right now in clinic. And give us an update on progress.
You bet. Thanks, Matt. So the most important one is called Keynote 695. And in cancer and cancer research, a drug called Keytruda has been approved. It was approved several years ago, and it's been approved in multiple different types of cancer, solid tumor cancer indications since. And it's an incredibly successful drug. It's an incredibly important drug. And, in oncology, it works about 30% of the time, which in oncology is good. Sometimes less, sometimes below 20%, sometimes 20%, sometimes 30%. The first approval Keytruda got was in first-line metastatic melanoma with a 24% response rate, meaning that 24% of the patients saw their tumors shrink by more than 30%, and no new tumors appeared. So that's kind of the industry standard to determine the criteria to determine whether or not the patient's getting a positive response from the drug, tumor response or tumor shrinkage. And so our study that we're doing is taking those patients who got Keytruda or another drug that's designed the same way. It's BMS's nivolumab or Opdivo. And, if you're watching TV, you'll see Ipi-Nivo, you'll see Opdivo, Yervoy commercials on or Keytruda commercials on. There's a fair amount of direct-to-consumer advertising going on with respect to these two drugs, cancer immunotherapies. Incredibly successful. But, you can imagine if in melanoma, it works about 30% of the time, meaning that 30% of the patients see the tumor shrink, the other 70 don't. And, and again, in oncology, 30% is actually a really nice response rate for patients who got metastatic cancer. On the other hand, what about the other 70? And, more importantly, what is it about those people that they got this important drug, this checkpoint inhibitor, Keytruda, Opdivo, and didn't see a benefit? And really, that's been a very important, pursuit for cancer researchers. And that's what OncoSec is engaged in. And we're not alone in that because, you can imagine, again, if you're able to make inroads in that majority of the patients that get the drug, but it doesn't help them, if you can change that and make it so that they get the drug and it does help them, then, that's actually doing something really good for those folks. And so what we're doing is we're taking a patient who got Keytruda or BMS's Opdivo, who have late stage metastatic melanoma, meaning skin cancer, that's stage 3, stage 4, which is the later stage, unfortunately, part of the disease. They get these two drugs and they don't see their tumor shrink. In fact, they can see them growing. And that's when they become eligible for our study. And so then they get our intratumoral, what we call TAVO, putting it right into the tumor. We don't have to treat every tumor. We can treat one or two or a couple, and we see a whole body effect, which is called an abscope effect. It's very important because if you're treating if you have to treat every tumor, then it's going to have limited utility, right? So we don't need to do that. We've shown that as what's known as a single agent monotherapy. But in this study, we take those patients who unfortunately got the Keytruda or Opdivo or both, didn't see a benefit, they come on to our study and get intratumoral IL-12 or Tavo, and then get Keytruda again. And, that's the study we're doing now. It's 100 patients. It's fully enrolled. The FDA has designated a fast track program and a U.S. orphan drug program. Mm-hmm. And Merck is our collaborator, partner on the study. And, it's obviously an important study because if we can, change that patient who got their drug and didn't see a benefit into having this intratumoral application occur with, the side effects I described, which so far have been, I think we've had 1 or 2 grade 3s, which is on the kind of risk benefit extremely favorable, and see their tumor shrink. That's a really, I think, a very important medical finding and an important thing for doctors who treat melanoma patients. But, to your point, it's not just that, because we view our drug as tumor agnostic. Just, like Merck's Keytruda works in lots of different solid tumor cancers, lung cancer, liver cancer, I don't know if they have an approval in liver actually, but lung, skin, head and neck, there's a kind of a large scope of approvals that Keytruda is gotten from FDA. Similarly, we don't think our drug is tied to a particular type of tumor like melanoma. It's a pro-inflammatory cytokine, which is going to work whether or not, or we believe it should have an effect if it's, whether it's melanoma or cervical cancer or head and neck cancer or Merkel cell cancer or, fill in the blank cancer. And so our goal is to really, draft off of Merck, if you will, where they have gotten approvals, go, and those patients that got their drug and didn't get a benefit, change that. Yeah. And give them this, relatively simple Tavo, intratumoral approach, and then give them Keytruda again. And hopefully you can bring back 20% or more of the patients who, previously got the drug and didn't get a benefit. So we're looking at that in breast cancer, triple negative breast cancer. And we're looking at a neoadjuvant melanoma, which means pre-surgery. Seeing if we can change the cancerous nature of the tumor before you surgically resect it. And which has, a medical benefit if you can kind of treat it very, very, very early. And again, our drug with the safety profile we've demonstrated so far really kind of directs you to the front of the treatment paradigm, because you're really not exposing people to, something you don't want to, but you have the potential to give them a nice, therapeutic effect. Yeah. There's another IND plan this year, which is an enhancement off of our plasmid. The plasmids are like blank slates. We code IL-12 onto it now, but we're planning to code something called IL-12 and CXCL9, a cytokine IL-12 and chemokine CXCL9. We think, and we've seen in our research and samples from patients on our studies, that we think the addition of CXCL9 to IL-12 can make even a better immune response and a better anti-tumor response. So that's a new IND we're planning to pursue this year, as well as what's called the visceral lesion applicator. Today, when we treat patients, we're treating only lesions, only tumors that are on the surface or near the surface of the skin. So within 1.5 centimeters, because that's the depth of the electrodes that are at the tip of our applicator. And so that's the depth we can go to reach a tumor. So it gets surface lesions or subsurface, like a lymphatic lesion, we can access. But we've miniaturized the technology so that it can fit through a trocar or a fixed needle or a catheter. We've taken it down to 1.9 millimeters. And then we can deploy, instead of, we have 6 electrodes, very small stainless steel, needles basically, to 2 electrodes. And when it deploys inside the tumor, inside the body, deeply inside the body, like a liver lesion or a lung lesion, we can kind of get to that location using imaging and then deliver, the plasmid and then electroporate. And that idea is really very, very innovative, because it would be in the hands of interventional radiologists who today are using ablative technologies to treat a tumor, right? They're burning, freezing, or, doing kind of a 1-for-1 tumor destructive, modality. Here, you're putting in their hands a medicine, an immunotherapy, something that actually can treat a piece of a tumor, but spread throughout the body, the whole body.
Yeah.
And sorry, go ahead.
No, I was just curious about, in that instance, how many, do you have any idea yet? Is it too early to say what sort of treatment frequency would be required?
Great question. Sure, you're not a doctor?
Well, no,
I
honestly,
that's a great question.
well, Dan, I bring it up, partly from personal curiosity. To be honest with you, I've been ferrying my dad to and fro down to Pittsburgh for treatment for bladder cancer. So your discussion around the catheterization aspect and being able to treat tumors deep inside the body is interesting to me. And, suffice it to say, the standard of care for bladder cancer is not a pleasant experience for man or woman to go through. So just curious about what the forecast for that might
and
look
the
like.
frequency is really a really important question, because you don't want to do that procedure, frequently if you don't need to. Yeah. And that's something we'll evaluate. I think, we right now, when we treat a patient, we treat on days 1, 5, and 8. So when you've got a skin lesion like melanoma or breast cancer lesion, which sometimes forms on the chest wall where the breast was previously before mastectomy, tumors can grow out from that location, and so that's accessible, right? Miniaturizing and when we do that, we do it on days 1, 5, and 8, and then we do that every 6 weeks. So the patient doesn't return until 6 weeks hence and goes through that same 1, 5, 8 treatment. We don't want to do that same level of frequency of treatment with a patient that's getting a catheter for a bladder cancer. But you could do it every 30 days, and this is one of the reasons why we added in a chemokine and CXCL9, and we actually moved from a higher energy system to a lower energy system, because when we use the lower energy system, we show a higher level of what's called transfection, which means the cell is making IL-12, because again, we force that DNA into the cell using energy, and then the cell starts to make IL-12 or CXCL9, whatever we engineer the plasmid or design the plasmid to make. So lower energy yields higher transfection, which means DNA moving from outside the cell into the cell, and expression, which means the cell's making IL-12 or making CXCL9. So we've shown preclinically that when you use lower energy, you get higher transfection expression rates. And that potentially could set up for your question, which is less frequency of treatment, right? And something like every 30 days.
Yeah. Yeah. Well, I feel as though we could spend a long time talking about this, Dan. It's fascinating science. It's great work that you're doing, but unfortunately, we're running short on time. So I'd ask you, and we can certainly have a to be continued, because I'd love to jump back on the phone with you, maybe as the clinical studies progress and we've got more news to share.
Thank you.
But to wrap things up for today, what would you kind of call the next big step for OncoSec?
Yeah, I think finishing the study I described, where patients got Keytruda or BMS's Opdivo and didn't get a benefit, get our drug, get Keytruda again. We've got that fully enrolled. We shared data the end of last year around the study, which is around the midpoint of the study. We liked where we were at, and we are looking to finish and file. Is the ultimate goal. That top-down approach, that's the goal. And that's probably the most important thing we've got going today. The other programs are very, very important to us, because it will be the next step beyond that, especially, moving inside the body. We also have engineered a COVID-19 vaccine, which I mentioned earlier, got permitted by the FDA to do a phase 1 study in the United States. Not a lot of vaccine candidates that fall into that category. So we consider ourselves very fortunate and privileged to be able to be looking at that to help solve that problem. And so that program's a pace. That could overshadow everything.
Sure.
Where that goes. But our focus is cancer immunology. And anyway, so hopefully that did that. I want to, before I forget, I want to say, I hope your dad and your dad's treatment go well.
Oh, thank you. Yeah, it actually is going quite well. It's going quite well. As a matter of fact, we were down there a couple of weeks ago for a checkup, and he's all clear. There is some routine maintenance that needs to take place for probably for the rest of his life. But at this point, he's all clear. He's in great hands down there. Dr. Benjamin Davies, a little shout out to him at UPMC. Awesome urologist doing great work. So thank you for your concern there, Dan.
I'm excited for you that he's in a good spot.
Yeah, for sure. Yeah. All right, Dan. Well, thanks again for joining us. It's been a real pleasure. And, I just, again, want to thank you for your lifetime dedication to service to others. It's very apparent, and I wish you well and keep up the great work.
Thanks, Matt. Like I said, when you give, you get, and so it's always just my pleasure, and I appreciate your time, and really again appreciate the time that you spent studying our company. It was definitely evident, great questions, and we appreciate that.
Yeah, well, we'll continue to pay attention.
Awesome, thanks so much. Have a great one.
So that's OncoSec's Dan O'Connor. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva, a company devoted to supporting new and emerging biotechs in deed and in action. Nowhere is their devotion more apparent than at the portal dedicated to the emerging biotech leader at cytiva.com/emerging biotech. That's CYTIVA.com/emerging biotech. Check that out. Check us out at bioprocessonline.com, where you can sign up for our newsletter, and give this podcast 5 stars if you liked it. Until next week, thanks for listening.